All right, thanks so much. Hopefully everyone can see the slides all right now. So Critical Path Institute is a nonprofit focused on supporting drug development, research and drug development research tooling. And they also host a rare disease data platform focused on breaking data silos, promoting data sharing, harmonization and aggregation of data, and generally maximizing the use and utility of rare disease data. So in general, promoting the FAIR principles in the context of rare disease research. So this is an illustration of the full data pipeline. So different data types such as clinical trial data, patient registries, natural history studies, et cetera, are ingested as inputs into the pipeline. They're archived and in some cases standardized or mapped into specific common data models. Metadata for the data sets is also curated and annotated before the data is uploaded to the platform, where it can be requested by external researchers or used internally by CPATH's own quantitative medicine programs. So the work that I'm going to describe is in this portion of the pipeline, where we want to annotate the dataset metadata, like the dataset or data dictionaries, with ontologies in order to promote internal search and discovery. So basically being able to find data sets that might be applicable for some use case. So the reason we want to do this annotation is to support semantic search and discovery on the left. So this is a very small subset of the hierarchy in the human phenotype ontology, HPO. And for example, we might not have variables or questions in our data that specifically reference recurrent maladaptive behavior as stated. But there's 120 subclasses there representing other concepts that might be more directly captured in the data that we're trying to catalog. So the idea would be we want to support a search platform where you can just look up maladaptive behaviors and immediately be pointed to the data sets and even down to the variable level where information about that term or one of its subclasses is described. The flip side of that is that then this also enables aggregation to sort of improve general visibility into the catalog. So we should be able to look at all these subclasses of something like a typical behavior and say how many data sets or how many variables do we have across the entire catalog that reference each one of those concepts. The way the annotations are materialized within the metadata knowledge graph is you have elements of data dictionaries captured as nodes, like you can see on the left. So a data set has tables, for example, a medical history table, which has columns, for example, a symptoms field. And then there could also be lookups that apply to or controlled vocabularies of specific columns. So maybe there's a lookup option like motor seizure within a symptoms field. Each one of those data dictionary elements can then be annotated with a corresponding ontology term. So in this case, the motor seizure lookup would be annotated with that term in the human phenotype ontology. And then using the part of relationships on the left and the subclass relationships on the right, we can infer and then materialize all the basically pairwise combinations, edges that appear above the original annotation. So the presence of those edges then in the catalog is what allows you to go to a term like seizure, the superclass, and quickly find out which data sets have some data that applies to it or one of its subclasses. This is an overview of the annotation pipeline as a whole. So data sets that might be in a common data model like study data tabulation, CDISC format, or non-standardized data sets or semi-standardized data sets with custom data dictionaries are all collected into the pipeline by extracting the text elements. So things like table names and table descriptions, field names, field descriptions. Those descriptions or names are pre-processed, and then a initial set of candidate annotations is produced using a tool, an open source tool called Fast HPOCR. So this is something that we found and are using in this pipeline. We found it very useful because it's a flexible methodology, but it's out of the box, works with Mondo and HPO, two of the ontologies that we're trying to produce annotations with. After that initial candidate annotation step, there's an LLM-based step to filter, subset those candidate annotations to try and improve precision. We're using SESM as the model for these annotations or schema, and I'll talk a little bit more about that in a minute. But those filtered annotations are then ingested into the catalog. This is an example of the framework that we're using for the filtering step of candidate annotations. So for both the examples in a few shot setting and the prompt structure, the way it works is the input is the text itself. So something like in the past 10 days, the patient's sleep quality was. So this might be the label of a question in a data set. And then the other half of the input is the annotation that FastHPOCR provided and the context around the term. In this case, the context is just the description of the term and then any synonyms that might apply as well. The output then is a classification of how appropriate that annotation was for the text. So basically the degree of relationship. So unrelated, somewhat related, all the way down to an exact syntactic and semantic match. As well as for transparency or explainability of the task, a reasoning description. So in this case, the model might generate that the text is discussing sleep quality, the annotations about narcolepsy, a specific disorder, so the degree is somewhat related. The purpose of this review step is to improve the precision by removing false positives. So some false positive sources that we see in the case of our data are things like acronyms. So feeding through text variability could be associated with the string fed, which is the same as an acronym for fisheye disease. Another instance is, of course, words with multiple meanings. So a word that has more than one meaning is false. In HPO, we're talking about it in the context of the subclass of gait disturbance. In this pipeline currently, so the LLM step is only used for reviewing and then filtering annotation, so it's not introducing any new information to the catalog. Something that I wanted to highlight is that we're using SESM, or the simple standard for sharing ontological mappings, as the internal schema for our annotations. So as the name implies, in general, this is a schema for sharing mappings between ontologies externally or with other groups. So we're mapping it from data dictionary elements, which have identifiers that are only meaningful internally to this pipeline. So these mappings we're not producing something that is shared externally, but we've still found that using the schema has been incredibly valuable because it's a defined standard with robust documentation and also some associated tooling. So like a Python package that you can use to validate SESM formatted metadata, et cetera. And the fact that we're using this defined schema enables us to build the pipeline in a more modular way. So we can put a wrapper around FastHPOCR set it outputs in SESM, and then myself and others can develop other pipeline steps that then are able to ingest those outputs without redoing any of the work of developing a schema like this. Another thing I wanted to point out about the pipeline is that because we're using these OVO ontologies as the vocabularies of choice to create these annotations over our data dictionaries, that also means that they become interoperable with external sources of knowledge like the Monarch knowledge graph. So this is a open source data set of biological association data from the Monarch initiative. There's a public API for retrieving these associations. So taking those things together, what that means is that we can put together catalog search applications like what I'm showing here, where you can input something like a Mondo disease term, build the concept set of all the phenotypes related to that disease, and then quickly visualize over the catalog how much data we have for each component of that concept set. So just a way to explore the catalog in the context of biological knowledge that's not actually present in the catalog or the data sets themselves. The next steps for this project then are, so far we've been validating the pipeline sort of in a qualitative way, looking at the annotations one at a time. Do these outputs for this data set make sense? Are these annotations useful? But we want to do this in a more quantitative way by putting together a gold standard annotation data set for some of these data dictionaries that will allow us to test the effect of different components in the pipeline, such as the examples used in the ViewShot prompt setting. It will also allow us to compare the current annotation tools we're using to new ones. So there's another open source tool that we're looking at called RAG-HPO, and this uses, my understanding is it uses a vector search to find those candidate mappings instead of string matching over an index. So by putting together the gold standard annotation data set, we'll be able to quantify what sort of improvement, if any, we see to our pipeline in the case of our data by swapping in newer tools like this. Thank you very much for your attention. I can take any questions, and I'm also happy to follow up over email as well. Thank you very much.